Hello everyone. Today our topic is gluconeogenesis. That is, glucose is produced in our body. Mainly we know that we consume exogenous glucose, that is coming through our diets. But this glucose is very important for our body. It is utilized in producing energy ATP, and remember, excess glucose is stored in the body. It is stored in the form of glycogen. It is converted into fatty acids. Okay, everything is happening, but every time we are not consuming food, but glucose is required continuously for certain cells like the brain cells. Then we have the retina, then the lens, then we also have the gonad cells, which are continuously requiring glucose for metabolism, right? So that glucose in the fasting stage or in the postprandial stages, that glucose has to be provided. Okay, now glucose is provided from different sources. Like there can be certain amino acids which can produce, there can be the fatty acids which will break down, that is triacylglycerol is there. Triacylglycerol will break into glycerol, and that glycerol will be used to produce glucose. So glucose is coming from carbohydrates, non-carbohydrate source, that is basically called as gluconeogenesis. Here we are seeing that the main site of gluconeogenesis is liver and also in the renal cortex, mainly in the liver. Why is it happening? Because of the presence of certain enzymes in the mitochondria as well as in the cytosol of your hepatocytes and the renal cortical cells. Talking about that, when is it required? It is mainly required in the fasting stage. Okay, because that time we are not consuming anything, but glucose supply has to be maintained. So body is using the glycogen reserves, the glycerol, and also the amino acids from which it is producing glucose. Okay, there are certain pathways that we will come across now in this lecture. Talking about from adipocytes, we have the triacylglycerol. This triacylglycerol is converted into glycerol. There will be hydrolysis and transport of this glycerol in from adipocytes to the liver. Okay, and in the liver this glycerol will be actually phosphorylated by the kinase enzyme. So it will be phosphorylated to glycerol 3-phosphate, and inside the liver kinase will act, and then this 3-phosphate will undergo dehydrogenase reaction, and dehydrogenase by glycerol 3-phosphate dehydrogenase, it will get converted into dihydroxyacetone phosphate, DHAP. Now remember DHAP, if you remember the steps of glycolysis, see, directly you cannot reverse that. You will see, you know that most of the steps of glycolysis are reversible, so it's not that you can reverse it anyhow. No, there is requirement, because there are 3 steps where they are irreversible, and for that you require certain different mechanisms. So one of them is that using DHAP, when you will form DHAP, it will get converted into glyceraldehyde 3-phosphate, then it will convert into fructose 1,6-bisphosphate, from there to fructose 6-phosphate, okay, then to glucose 6-phosphate and back to glucose. Like this the process will happen. Okay, so this is an important step, that from glycerol how we are getting your glucose. Then talking about in the muscle cells and in the red blood cells, we are seeing there is an aerobic oxidation of glucose, that is producing lactic acid. Okay, pyruvate is produced, and from pyruvate lactic acid is produced. Now this lactic acid is transported back into liver, and there it is oxidized by lactate dehydrogenase into pyruvic acid. Now this pyruvic acid there forms inside the liver, it forms glucose, and that glucose is again transported into the liver, into the muscles via RBCs. Okay, so that is, this cycle is called as the Cody's cycle. Now talking about that, what actually happens with this alpha keto acids like the pyruvic acid, and from how are we getting back, because you know that phosphoenol pyruvate, the last step from phosphoenol pyruvate to pyruvate, we are seeing that reaction is irreversible. So from pyruvate we cannot directly go back into phosphoenol pyruvate. For that we require certain series of steps, and that is happening in the liver cells. Okay, now see, the mitochondria of the liver, we have pyruvate present. Now there will be the action of enzyme carboxylase. Carboxylase will act, and this carboxylase is basically, it is carboxylation, so carbon dioxide is required. So this carbon dioxide is produced by bicarbonate source, and from bicarbonate carbon dioxide is incorporated. Now here is requirement of vitamin B7, that is biotin is required. There are epsilon amino acid bonds actually present in the lysine residue of this enzyme pyruvate carboxylase, where this biotin gets attached. and from pyruvate we will see the formation of oxaloacetate. You remember oxaloacetate in the TCA cycle we have studied. So from this oxaloacetate produced in the mitochondria cannot get transported into the cytosol. Okay, so it has to be converted into some other forms like malate. Malic acid is produced by dehydrogenase. This is a type of malate dehydrogenase in the mitochondrial variety. Now it will convert into malic acid to malate, and this has certain specific transporters which will help it to get transported into the cytosol. Now inside the cytosol, you remember again this has to be converted by cytosolic malate dehydrogenase into oxaloacetate. This reaction has to take place in a reverse fashion, okay, a bit in a twisted way. Now see again it is getting converted into oxaloacetate, and this oxaloacetate, now it is the time for this oxaloacetate to get converted into phosphoenol pyruvate. Now oxaloacetate to phosphoenol pyruvate. Now this is a decarboxylation reaction. Here the carbon dioxide will be removed, and also this required reaction, as I have told you, the enzyme is carboxykinase. So whenever kinase will be there, remember some phosphate bonds will be utilized, hydrolyzed. So here in this case GTP, guanosine triphosphate, that from there the phosphate group will be taken, and here it will be attached to the phosphoenol pyruvate form. Now from phosphoenol pyruvate you can reverse the steps of glycolysis. You will reach a point that is the fructose 1,6 bisphosphate stage. Now in this fructose 1,6 bisphosphate, now here comes the action of bisphosphatase enzyme. Okay, here because this was an irreversible step, so here you require the bisphosphatase enzyme which will convert it into fructose 6 phosphate by removing one phosphate group. And from fructose 6 phosphate again you can isomerize it to glucose 6 phosphate. As we had already seen, there was the enzyme isomerase which used to isomerize them. And from glucose 6 phosphate, you remember from glucose you get glucose 6 phosphate by hexokinase, and when there will be glucose in excess, glucokinase will act. So here again phosphatase will act, and glucose 6 phosphate will get converted into glucose. Okay, so remember this bisphosphatase or phosphatase, these are stimulated by glucagon. Okay, the regulation we will study, remember that mostly we require glucose, new glucose in our body during fasting stage, because whenever fasting is happening, if it is less than 24 hours, then from glycogen reserves there will be glycogenolysis, and from there we will get to glucose. Okay, but when we are actually fasting for more than 24 hours, that point of time these amino acids will be utilized, or there will be other metabolites they will be utilized to produce glucose, because that time glycerol reserves will be all not glycerol, that is your glycogen reserves will be consumed. Now as I have told you that here we require glucagon, which is the key enzyme of during the fasting stage. Glucagon is the main enzyme, so glucagon will promote gluconeogenesis. Here we see that two most important things, one is AMP, adenosine monophosphate, and fructose 2,6 bisphosphate. These two substances, they will inhibit the phosphatase, whereas glucagon will actually activate the phosphatase or the bisphosphatase, whatever. So talking about the regulation, what will happen whenever glucagon level will increase, that is during the fasting state glucagon level is increasing, so under influence it is stimulating the adenyl cyclase, which is converting ATP into cyclic AMP. Okay, ATP is actually converted into cyclic AMP with the enzyme, that is adenyl cyclase. Okay, now cyclic AMP's concentration is increasing, so cyclic AMP has that tendency. So this phosphate, where is this phosphate going? Phosphate is actually phosphorylating certain enzymes. Okay, one of the enzymes is the bifunctional enzyme phosphofructokinase, and also fructose 2,6 bisphosphatase. These enzymes are basically activated or inactivated. They are inactivated by phosphorylation. Okay, phosphorylation is happening and they are getting inactivated. This phosphofructokinase 2, this will first get inactivated. So when this will get inactivated, glucagon will already act on fructose 2,6 bisphosphate. Glucagon will act on fructose 2,6 bisphosphate. It will decrease the level of hepatic fructose 2,6 bisphosphate. Okay, and you know that whenever AMP's amount is increasing, So when AMP's amount is increasing, that has a tendency to also inhibit fructose 1,6 bisphosphatase. As I told you, here also fructose this 1,6 bisphosphatase enzyme was inhibited, okay, by AMP. So AMP will have this tendency to inhibit that. So that cannot be allowed, okay, because we have to promote gluconeogenesis. In that condition, what will happen? In that condition, the fructose 2,6 bisphosphatase domain will actually become inactivated, okay, that will become inactivated. Phosphofructokinase was inactivated, because see, when phosphofructokinase is inactivated, so automatically that cannot promote glycolysis. This will favor gluconeogenesis only, and at that point of time, this fructose 2,6 bisphosphatase, that enzyme will also get inactivated. Okay, in special cases there will be certain certain locking and unlocking mechanisms are actually there, that how this is activating this, and there is a cascade of quite complex reactions. But remember, for exam purposes, when we write that how glucagon will actually activate the protein kinase A, and at times also it can activate the G protein coupled receptors. They will be activated, that will activate protein kinase A, that will increase level of cyclic AMP, and like this phosphorylation of the bifunctional enzymes will happen, and when they will be phosphorylated, they will get inactivated, and when they will get inactivated, this will promote the bisphosphatase enzyme. Glucagon will promote the bisphosphatase enzyme, and fructose 1,6 bisphosphatase will be activated, so that fructose 1,6 bisphosphate is converted into fructose 6-phosphate. From there it is converted into glucose. So gluconeogenesis, as I have discussed, that you see that from pyruvate mainly, because we are seeing from glycolysis that one glucose produces 2 molecules of pyruvate. So here 2 molecules of pyruvate will produce 1 molecule of glucose. In this case, in the total reaction, we will find that total 6 phosphate bonds will be cleaved, high energy phosphate bonds will be cleaved, and this NADH, that will be oxidized into NAD plus. Okay, NADH will be oxidized into NAD plus. Total 2 molecules of, overall I am talking about the overall reaction, that will be favored. So that was about, in short, I have discussed gluconeogenesis. Mainly remember that whenever in the fasting stage, kidneys will actually be more activated comparatively. Okay. Another important thing, remember that why does these things happen? Because in the mitochondria you will find the p-carboxylase, that is pyruvate carboxylase, is present in the cytosol, in the mitochondria of liver and kidney cortical cells, and that's why gluconeogenesis is happening over there. Another important thing, that acetyl coenzyme A, whenever there will be higher concentrations of acetyl coenzyme A, that will stimulate this carboxylase. That is, when acetyl coenzyme A level will increase, that time it has a higher tendency of promoting this reaction. Okay, that will happen. Whenever we will study integrated metabolism, that point of time we will know that how different pathways are inactivated at certain points and how others are activated, so that body can maintain its regular dynamic state. Okay, it can function properly. So that has to be regulated. So this was all about gluconeogenesis in short. Thank you.